Okay, so for this audio lecture, we're going to be looking at immunity mediated by B cells and antibodies. So this is also referred to as humoral immunity, so immunity in the fluid because antibodies get secreted and are found in the fluid. In order to have antibody production, we need to have B cell activation. So you're going to have your B cell after development, it's going to have its B cell receptor, and activation of the B cell can occur either in two methods where we have T-dependent response. This stands for thymus dependent. And so with thymus dependent response, this is where T cells lend help. So you remember those T follicular cells. Those are going to help the B cell so that it can become activated. So with T-dependent activation of B cells, this is usually with protein antigens. So it's usually the method that's utilized. They require, for protein antigens, usually you require T cell help in order for the B cell to respond to it. There's going to be three signals for T-dependent activation. So the B cell receptor is going to be interacting with the antigen. But then you're also going to have MHC class 2s on the B cell that's going to be interacting with the T cell receptor. So you're going to have a complex forming between the T cell and the B cell. You're also going to have complex happening between the B cell receptor and the antigen, and then the third signal being cytokines. So it's not the classic, when we're talking about three signals, usually that we're talking about T cells, but to just kind of do similarities, again, think of B cells when they receive T cell help, they kind of have three signals too. The T cell help allows for the B cell to undergo class switching and also somatic hypermutation. So because of these interactions with the T cell, there's cell signaling events that are going to happen that allow for class switching to occur so that that B cell is going to change the constant region that's being expressed. And then it also can do the editing for somatic hypermutation, so activating enzymes like AID to allow for the editing to occur. So the other way that B cells can become activated is T-independent. So T-independent is thymus independent, so this does not require T cell help. So for this, usually what's happening is that we have cross-linking with the B cell receptor with antigen that's still on the bacterial cell. It's also possible that through interactions with the B cell receptor and also pattern recognition receptors, this can also lead to activation of the B cell. So there's what's known as TI1, so T-independent 1, that's usually through pattern recognition receptors, and TI2 is through the B cell receptor, but because the B cell receptor complexes together, there's multiple interactions with antigen and B cell receptors on the B cell that leads to the signaling events for it to be activated. So we're actually going to look at T-independent responses first, then we'll look at T-dependent responses. So when we're looking at the independent response, here we see the B cell receptor and it's interacting with antigen. Because each B cell receptor is going to have two binding sites, it can have multivalent binding. Again, these receptors usually complex together, so it's not just one interaction, there's going to be multiple interactions that happen. And like T cells, B cell receptors are going to have ITM domains, so these ITM domains end up getting phosphorylated through kinases that are nearby, such as LINC, BLAC, FLINC, and SYK. And so again, when these get phosphorylated, this then causes downstream activation of cell signaling pathways. So you get changes of gene expression and cytokine production, so that you get activation of humoral immunity. Keep in mind that with T-independent response, again, you classically don't get class switching and you don't get somatic hypermutation. So these aren't necessarily like ideal antibodies that you're going to produce, but at least something is better than nothing. And it gives you an initial response. Just remember that with the B cell receptor, there's co-receptors. So a little bit different than CD4 and CD8. It has CR2. So you can see how CR2 will interact can interact directly with the pathogen, allowing kind of for an anchoring. We'll also see in our next slide how it can allow for stabilization so that the B cell receptor can interact with those antigens that are on the bacterial cell. If there is soluble antigen, so if there's antigen in circulation in fluid, you can see how C3d binds to it, and then that allows for CR2 to bind that and basically hold it in place. So it's going to interact with those antigen binding sites on the B cell receptor, and then allow for phosphorylation events that activate cell signaling pathways and changes in gene expression. So again, you can see that in that left figure. So on the right hand side, this figure is just kind of emphasizing that just keep in mind that this isn't the way that all B cells get activated. So this is only for thymus independent activation. So this is antigen that has not been processed and presented to T cells. Okay. And this doesn't give you, again, a strong response, but again, it gives you some response. So these naive B cells, they can also interact with antigen as they're entering into secondary lymphatic organs. So there will be macrophages that have taken up antigen, and so they can show that to the B cell, and the B cell could respond to it if it recognizes it. So if the B cell receptor recognizes it, they can become activated in that way. They can also enter into the follicle, and if there's a follicular dendritic cell that is presented that has antigen to show, again, the B cell could interact with that. So both these ways, whether it's macrophages or dendritic cells, again, these at this point would still be considered T independent activation. So you're only going to be getting the one signal from the B cell receptor. And so again, the response that you would generate isn't going to be as robust as if you have three signals, like when you have T cell help. So now we're looking at T cell dependent helps or T dependent help. So here we have our T follicular cells. So if looking at this figure, we have naive B cells and T cells coming into the secondary lymphatic tissue through the HEV, so high endothelial venule, and you can see how the T cells, like we talked about last chapter, those naive T cells are going to interact with dendritic cells. So if they end up seeing the antigen that dendritic cell has, they're going to start to proliferate. You'll notice that the naive B cells move to the primary follicle so they can interact with the follicular dendritic cells, like we were just talking. If they see the antigen, they're going to end up moving to the boundary region. And this is right on the edge of where the T cell zone is. And so this allows for the T cell and B cells to interact with each other. So you can see that on the right hand of this figure, where the B cells have moved to the boundary region and the T cells also move. So they both have seen their antigen, and so they can try to form what's called cognate interactions, cognate pairs. Cognate being that they see the same antigen. They may not see the same exact epitope because they're not going to be able to bind the same spot at the same time. So if we look at this in a little more detail, in this figure we can see how that T follicular cell is interacting with the B cell. So you're going to have lymph1 expression that is going to interact with ICAM. So ICAM1 is going to be expressed by the B cell. Lymph1 is expressed by the follicular T cell. So that's going to kind of dock. Again, you can think of these cells as kind of docking together. Then we're also going to have interactions between the T cell receptor and the MHC class II. So keep in mind that on the B cell receptor, you can see how it's bound to antigen. That antigen is also being presented by the B cell to the T cell, right? On the MHC class II, T cell receptor recognizes that. And then we're also going to have co-stimulatory molecules. So we're going to have CD40 being expressed by the B cell and CD40 ligand by the T cell. So those are going to interact with each other. So again, we can think of this as three signals. So first signal is where we have the B cell receptor binding to antigen. And then we have the interactions between the MHC class 2 and with the T cell receptor and its co-receptors. And then we have CD40 ligand as our co-stimulatory. And then lastly, we have our cytokines. So cytokines are going to be secreted by the T follicular cell, which is then going to give that kind of third signal to the B cell so that it can activate in a T cell dependent manner. And so you can see in this image, on the image on the right, looking at microscopy images of the T cell and B cell interacting with each other in the kind of contact that's going to be occurring between them. So keep in mind when the B cells are becoming activated, they're kind of making this, they're coming in, they're going to the boundary zone, they're interacting with the T cells. And what they'll do is the pair will actually move to the medullary cords, where there's going to be cues and signals that are going to occur. And then what happens is the B cell kind of takes the T cell back into the T cell zone, and the B cell is going to continue and create a germinal center. So again, you kind of think this is like a dance. It goes from the follicle to the boundary region to the medullary cords, then back out to the follicle where it's going to create a germinal center. So the follicle is more of like a loose kind of association of these cells, where with a germinal center, this is where these cells start to grow. They get, the proliferation is occurring, so they're increasing in number, so it becomes very densely packed there. And it's in the germinal center where the B cell can undergo affinity maturation, somatic hypermutations, and also class switching or isotype switching of their antibodies. So the kind of fine-tuning and improving of the antibody is going to happen in that germinal center. So here we have our germinal center. So you can kind of pick your image and figure which one is a little bit easier to see. If you look at the top images, I think those are probably the best ones because they have a little added detail. So you don't have to be a histologist to appreciate them. But you'll notice that there's certain zones and areas with our germinal center. So of course, we're going to have the T cell area zone around that. But then we have our mantle zone, and in our mantle zone is going to be up in the top portion. We have our light zone. Now in the light zone, we have some of these centrocytes, which are a stage of B cell, and we have our follicular dendritic cells. In our dark zone, this is where we're going to have centroblasts and our we're going to have some T follicular cells that are going to be able to interact with these different zones. So you see these T follicular cells around the outside. And again, they're going to be able to interact with these different zones on the boundary, on the outside of it, and provide cytokines that these maturing B cells would need in order to undergo affinity maturation and isotype switching. So remember, with somatic hypermutations or affinity maturation, this is where point mutations are going to occur in the heavy and light chain of the B cell receptor. This is done by the enzyme AID. And again, we talked about this when we were talking about the receptor, so that's unit 3. And the idea is that you're going to be selecting for higher affinity B cells. So the lower affinity B cells actually get, they don't get survival signals, so they're not going to be able to survive. But the higher affinity B cells are actually going to be able to bind antigen because they're higher affinity, and so they're going to get the survival signals. So here you can see, our two possible outcomes, right? So we have our activated B cell because it's received help from the T follicular cell. That's going to cause somatic hypermutation. And so we're going to have little changes, and that changes those B cell receptors so that we really end up with a left and a right, right? So we end up with some that are low affinity. And so you can see with the low affinity, it's not going to be able to bind that antigen well. It's going to get out competed for any of that antigen. And so again, it's not going to get the survival signals. So it'll end up going to apoptosis, as we'll see in the second part of this figure. If we have a higher affinity, which we're seeing on this like right side, it is going to be able to bind to, its B cell receptor is going to be able to bind to that antigen more strongly at a higher affinity. And so it's going to get the cues. It's going to also be able to interact with the T follicular cell because it's bound its antigen. So it's going to have signal 1, signal 2, and we're going to have cytokines present. And so it's going to get those 3 signals so that it can activate cell signaling pathways. And that will end up causing this cell, which is actually a centrocyte, to become a plasma cell. So you can see here on this second part of that figure, the kind of the outcomes of that, right? So you can see on the left-hand side that low affinity antibody due to somatic hypermutation, because these are just random mutations. So it could lead to a lower affinity or it could lead to a higher affinity. But if it's a lower affinity, it's going to lead to that B cell, the centrocyte, to die. Okay. If it's higher affinity, again, that centrocyte receives signaling cues to proliferate, to divide, to survive, to divide. And they can also differentiate into plasma cells, and plasma cells would be secreting antibody. This is just giving you some additional information. Again, with those germinal center cells, the idea is that they're going to end up maturing. The goal is for them to mature into plasma cells, so they're secreting antibodies. This is going to take around 10 days for this to occur. And this is due to interactions between the T follicular cells that are going to be around that germinal center. And this there's really important interactions that happen with the PD-1 receptor on the T cell and then the ligands on that are on the B cell. There's also going to be chemokines and cytokines that are important in homing these plasma cells back to the bone marrow, where they can become long-lived plasma cells. This is a type of memory cell that the B cell can produce. Now, as far as how the B cell knows which class of antibody to isotype switch to, it's because of the cytokine environment. So different classes of antibodies have different functions. So if you have different cytokines that are generated during a response to different pathogens, these cytokines then tell the B cell, let's produce the best suited class for that type of infection or immune response, right? So similar to what we see with the T cells and the different subsets, we can think of the antibodies as being kind of subsets of the B cells, depending upon what class they produce. And so when we have certain cytokines like IL-4, that's going to produce like IgEs because those are used in parasitic infections, okay? And so again, if you look at these different cytokines, they're going to lead to switching of different isotypes. So keep in mind when we have isotype switching or class switching, it is a recombination event. So AID, that enzyme, is going to come in and help in recombination, cutting out the constant regions that are not going to be utilized, so that you get the constant region in the correct position that's going to be utilized to produce the class of antibody that you need to respond to that. And so again, cytokines, so important. And so you can see here how that T follicular cell is producing cytokines, producing signals, so that it's going to enable this B cell, this centrocyte, to be able to differentiate into either a plasma cell or a memory cell. Again, keep in mind, plasma cells are going to be secreting antibodies. Memory cells are going to be able to differentiate into different types of B cells, but they can be recalled, right? So they can be recalled and then differentiate into plasma cells if needed. And so when we look at like secondary infections or secondary responses, this is why that memory population is so important. Or someone who's been vaccinated, they would have a memory population to call upon to quickly produce plasma cells, opposed to having to go through this whole process again. Because again, keep in mind, germinal center, that's like a 10-day development And so that's 10 days you're not producing antibodies to respond to an infection that's going on, because you know the infection is going on because you had dendritic cells presenting the antigens for that, right? So again, that ability to produce memory is really important, both for T cells and B cells. Number of different markers that we can look at as far as in properties between differences if we were to compare and contrast naive B cells and plasma cells. So your naive B cell is going to have surface Ig or surface B cell receptors, where the plasma cell is going to secrete that, right? So one of these intrinsic properties, one of these things that we look at to characterize, if we have secretion of the B cell receptor of the antibody, that tells us we're dealing with a plasma cell. Where if it is surface Ig and it has surface MHC class 2, that tells us it's a naive cell, right? So after the B cell has become activated, it doesn't need to interact with the T cell again, so it doesn't need to be able to present to it. So it can lose that ability to have surface MHC class 2. There's also inducible properties, so think about like making them behave a certain way. And so with a naive B cell, we want it to grow. We want it to undergo somatic hypermutation, isotype switching, because it's naive, right? So we want to develop it. Now, a plasma cell has undergone its somatic hypermutation. It's been proven to be a good high-affinity mutation, so we don't want to go through additional edits because we could go in the other direction. And we don't want to go through further isotype switching because we switch to meet the demands, the needs of the organism, of the host, to protect it. So it's good that we have that class. Let some other B cell go and produce a different isotype. All right, so now we're going to kind of run through the different antibodies and a little bit of their functions. And some of this, it has been covered in our previous units, but kind of just expanding upon it. And the big piece that we're expanding upon is Fc receptors. So there are Fc receptors. Those are able to bind the Fc region of antibodies. So keep in mind that if you were to take a protease and cut your antibodies, you would have two FAB regions, FAB, and those are the antigen binding regions. They would have the binding sites on those. The constant piece is the Fc, so that is that bottom portion where you have the constant region of those antibodies. And remember, the constant region is the one that's unique for each class. So there are receptors that can bind to that region. So here you see on these endothelial cells, they have Fc receptors. And so when you have IgG, it's going to be able to bind to those Fc receptors so it can be transported into the extracellular space. So this is how antibodies are going to get past cell layers. So things like endothelial layers and epithelial layers. So we need, in order for this antibody to be able to bind to whatever's causing the infection, whatever that recognizes its antigen, sometimes it needs to get into the extracellular space or it needs to get out to the lumen. Well, this one's in a capillary and it needs to get to the other side to kind of see what's there. And so the way it does that is by transport through the endothelial cell. I will say this image is a little wonky because you don't actually see the endothelial cells membranes, and that's a huge vesicle for a relatively small cell, but I digress. Okay, so again, all we're doing is endocytosis and then exocytosis, secreting it out on the other side. And then for IgA antibodies, really important for mucosal immunity, and so in order to get them into that mucosal layer, it needs to be transported from the basement membrane side, the basal side of the epithelial cells onto the lumen side, right? So if we think of like the gut is going to have this epithelial layer. That epithelial layer is going to have tight junctions. So it's not that the antibodies can squeeze in between the cells. If they tried to do that, they'd hit the tight junction and they wouldn't be able to do that. So they need to enter through that basolateral surface, which is the bottom surface. So here we see pig R, what we call PIGR. And so this is a poly Ig receptor. So it's able to bind IgA. So you can see how IgA is going to bind it. That causes endocytosis of the IgA antibodies. It's going to transport to the apical surface. So the apical surface is going to be the top of the endothelial layer to the lumen. And so it can be transported out. And again, keep in mind that there would be a mucosal layer here. And so now you have these IgA antibodies in that mucosal layer where they can interact if there's any kind of pathogen, parasite that is potentially disrupting that mucosal environment there. But again, the whole goal here is we've got to get the antibody to the other side so it can actually function. And so we're going to transport through the cell, not around the cell, because these are tissues just aren't going to allow for the separation of the cell for that kind of transport. And then when we look at IgGs are really important with mast cells. So you can see our inactivated mast cells, you can see those granules, those circles. Those granules are going to have histamine, pro-inflammatory mediators, so lots of different chemicals. So I think it's estimated that there's over 200 different chemicals that are found in the granules of mast cells. So when they degranulate, when they become activated, they release all of this. And these different histamine and different chemicals end up playing a role in inflammation and the immune response that would be generated during this. And the most classic one that we'd recognize is allergic reactions. So mast cells degranulate, release histamine. Histamine acts on a number of different tissues. So everything from the respiratory, which causes increased mucus production, causes your glands to secrete mucus and your tear ducts to secrete. So you'll get runny eyes. It can lead to hives. So the histamine release causes hives on your skin. And then you can also have a number of GI distress issues because these mast cells are dumping large amounts of pro-inflammatory mediators. And so you have all this inflammation in your gut too. And so you get those symptoms. You can have narrowing of the airways. So you can get, respiratory distress from this. And keep in mind, when we're looking at allergic reactions, we recognize anaphylactic reactions are severe. Traditionally, medical community views the breathing issue as an anaphylactic reaction, but anaphylactics can present in other ways. And so in our last unit, at the end of the semester, if we end up talking about allergies, we'll make sure we talk about anaphylactic reactions and how they actually do present in a number of different ways. It's not just a respiratory presentation. And so, but again, going back to the mast cells, you can see how they have it's degranulated, it's released that. And that happens when the Fc receptor on them binds to the IgE and the IgG has bound its antigen, right? So whatever this IgE antibody recognizes on its antigen binding site, when it binds to that antigen, that's going to cause cell signaling events through the Fc receptor to then degranulate and release these pro-inflammatory mediators, including histamine. The other thing that we see is that IgEs can, they'll recognize parasitic surface antigens. So this is what we're seeing here. It's recognizing the larval stage of a parasite. And so the antigen binding sites on the IgE are bound. And then in this case, usually it's going to be like an eosinophil that it is binding its Fc receptors to. That causes degranulation of the eosinophils. Eosinophils produce chemicals that paralyze these parasitic organisms, which give the immune system a better opportunity to attack it. It also leads to, if it's something like a parasitic worm that uses a hooker or a sucker to attach, they're paralyzed so they can't stay attached, and so they can be passed through the digestive system more freely when they're not attached, and that gets rid of them. So these eosinophils have saved their host. One of the things to keep in mind is that during different stages of life, different antibodies can pass through different tissues. So this is just looking at a healthy woman, and you can see how there's going to be certain regions where we would find certain isotypes of antibodies. So you can see in those mucosal surfaces, we have IgA. In the heart, we would actually find in circulation, which is demonstrated by the heart, we would actually find a number of different antibodies present, such as the monomer of IgA, IgG, and IgM. And then largely we think of, again, mast cells being located on the skin. And so you can see the outline of the skin where we have IgEs present, where they could interact with mast cells. Mast cells are also found in the gut and in the respiratory system, so you can also see those areas are also lined with IgEs. So again, we tend to focus on mast cells being on the skin because that's what we can see. We can see hives. But if you have degranulation on the skin, unless it's due to skin contact, but there's a good chance that you also have degranulation in your GI system and also in your respiratory system. And then when we look at in utero, we can see that IgG isotypes are actually able to cross the placenta and get to the baby. And then IgAs end up being secreted in breast milk. And so this is one of the reasons why women are encouraged, if they're breastfeeding, to continue to breastfeed even if they can when they're ill, because they can be passing antibodies on to the baby that would actually provide it with some protection, especially if it's young and hasn't had a chance to develop its immune system yet. One of the properties of some antibodies is that they can be neutralizing. So in these two figures, we're looking at the neutralizing effect of antibodies. And you'll notice that the examples they're giving are IgAs, but other antibodies, other isotypes besides IgA can have neutralizing effect. And so you can see with neutralizing effect, if we have a viral infection, they're going to block the glycoproteins, the surface proteins of the virus. And so that way the virus can't bind to the receptors on the surface of the host cell. And so we have this immunized individual and a non-immunized individual. An immunized individual has antibodies against it, neutralizing antibodies, and so it's going to bind to the influenza virus, so the influenza virus can't infect, so that individual stays healthy. Where the other one that did not get immunized, it goes and is able to bind to the receptor, the virus enters into the cell, it's able to replicate, and then that individual would become sick. Okay. In our other panel, we have bacterial infections. So there's a number of bacteria that produce toxins, and these toxins kill cells and damage these cells, and that's how the bacteria gains an upper hand on the host. And so here we can see how if we have an individual that has antibodies against streptococcus, they are going to be able to neutralize these toxins, and they're just going to pass through the gut, so that child remains healthy. However, if we had an individual that did not have antibodies against it, this protein F, this toxin that's being produced, is going to lead to damage in the cell. And so what you're seeing here is this kind of, breaking down of the tissues that are making up the pharynx. They're also going to the bacteria is going to be able to multiply, so it's going to get a hold on an infection, and so this individual ends up getting a sore throat from that streptococcus infection. But again, there's a number of other, besides streptococcus, there's a number of different bacteria that do produce toxins, and so some of the treatments, if you have an exposure, would be an antitoxin. So these antitoxins are usually antibodies that are able to neutralize it. Also, the goal when we're looking at vaccine development is to have individuals developing neutralizing antibodies. So that was one big part with the COVID vaccine, even though it's a virus, not bacteria. They were looking at neutralizing antibodies being produced in the vaccinated individuals versus natural infection. and vaccinated individuals had a higher percentage of neutralizing antibodies compared to non-vaccinated individuals, but who had natural immunity. So, but again, you can see here pick your favorite pathogen, your favorite bacteria, whether it's cholera or whether it's Clostridium. There's a couple different Clostridiums on there, but if they produce toxins, again, if you can neutralize them, they're not going to have an effect. So if you look at the effect, a lot of these are to inhibit some kind of normal pathway, whether it's a normal, like tetanus toxin is going to inhibit neuron action. So you have locking of muscle contraction. And so again, if you have an antibody that neutralizes that toxin, then it's not going to be able to have that effect. So here we can see that neutralizing effect of the toxin by IgG. So again, it's not just IgAs, IgGs can also neutralize. And so here we're going to have binding of our toxin so that, again, it can't bind to the surface receptor, which then allows it to act. IgMs and also IgGs are also known for their role in initiating the classical pathway of the complement activation. So if you think back to Unit 2, when we talked about the complement pathway, we mentioned that there's a classical pathway that's initiated through antibody binding or C-reactive protein, right? That's kind of added in, right? But if we're talking about antibody binding, then this could be an IgM or an IgG. So IgG3 are also good at activating the classical complement pathway. So they're going to bind to antigen on the surface of the pathogen. And then that's going to recruit in the C1 complex, activate C2, C4, activate C3. And then we're going to form MAC. We're going to activate C5, and then we're going to form MAC, the membrane attack complex. So kind of jumping back and looking at these Fc receptors, so we looked at a couple of these Fc receptors, but there are Fc receptors on macrophages and also natural killer cells. And so when we look at Fc receptors on macrophages, it can lead to phagocytosis. So keep in mind that this antibody the antibody's Fc region is going to bind to the Fc receptor. Its Fab portion, its antigen binding portion, is going to bind to antigen, and then this is going to initiate phagocytosis to occur. Natural killer cells also have Fc receptors. So they have an Fc gamma R3 receptor, and they're able to bind antibody. And so what individuals have done is they have designed monoclonal antibodies that bind to tumors specifically, and that leads to the natural killer cell being able to bind through their Fc receptor, and then that causes the natural killer cell to induce death. You can see here down at the bottom a number of different Fc receptors. A lot of them go by CD numbers too, so you might see one or the other. You'll notice that they look a little bit different. They can have ITAM domains to initiate cell signaling pathways that can lead to activation or inhibition. And this is a little more complete on these Fc receptors and what class of antibody binds to them. And keep in mind the different classes of antibodies. These Fc receptors can be found on different immune cells, which then means the antibodies are binding, causing, when the antibodies bind to these Fc receptors, it causes a different response from that cell. Okay, so again, just kind of keeping our example of mast cells and basophils. When we look at the FcRn receptor, this is going to be an activating receptor. And so when IgEs bind to it, and this receptor is found on mast cells and basophils, again, what mast cells and basophils do is they release histamine and all these pro-inflammatory mediators, and so that would be the effect you'd end up having. Okay. When we look at, for instance, IgA, which has its kind of distinct family for its receptor, For the Fc alpha R2 receptor, which is found on macrophages and neutrophils and dendritic cells, this would lead to phagocytosis when the IgA receptor binds to the Fc receptor. Again, you can go through the whole chart and look at that. But again, you can see how these Fc receptors are found on different cells. Very common to find them on macrophages. And keep in mind, they're not expressing just one single Fc receptor. They want to be able to interact with different types of classes of antibodies. And so they'll express multiple different receptors so that they can do that. And again, each one of them might generate a different response, different cytokines being produced, so that they can that cell can further the immune response correctly to that target pathogen. All right, and as always, if you have any questions, please don't hesitate to contact me. We'll be going over some of this in a little more detail in class, and we'll do some class activities also. So thank you for your time.